So let me give you an example of a project that we had in the past in which we used the knowledge on the biological networks that is collected in the field of biology to improve analysis of one of the most important data type in cancer research, which are mutational data. So a quick reminder to you about what is meant by somatic mutations in cancer. You know that each normal cell has a copy of DNA, and this DNA is replicated in the process of cell division, and this replication isn't perfect. So each cell division might introduce some mistakes in the copy of DNA. So and these mistakes start to accumulate during the human life. So in principle, those cells which exist in the human body, but in the adulthood, they already accumulated a certain number of mutations, and these mutations might be different from one cell to another cell. So this is totally normal process, which can be affected by some factors from the environment or lifestyle. And there is a so-called normal physiological rate of accumulation of mutations in dividing cells. But this process under some circumstances might be accelerated, and this is what typically happens in cancer. So one of the mutations might lead to abnormal proliferation of a cell, which in turn might increase the number of mutations, and at some point you might have mutations in a gene which serve as a genome gatekeeper. So which function of this gene is responsible for kind of conserving the genome sequence. And starting from this point, the process of accumulation of mutations might significantly accelerate, which lead to early invasive cancer, late invasive cancer, and then at some point cancer might be treated by, for example, chemotherapy, which also affect DNA. And in the end, so in the worst scenario, the cancer is developing the tumoral cells. They are converted into those cells which are chemotherapy resistant, so they don't answer as a therapy. And at each stage of this hypothetical cancer progression, the number of mutations which are accumulated during each cell division increases by some orders of magnitude. So this is a kind of simplified representation of the process of accumulated somatic mutations. And in principle, today, we take a tumor, we can have access to this information. We can determine which genes in the genome have accumulated mutations. And in the most simple case, we can say we can distinguish if a particular gene in a tumoral cell or a group of cells contains a particular mutation or not. So we can represent this information in the form of a binary matrix, which would contain as rows different patients or different tumors, and as columns different genes. So if in this matrix you have one at the intersection of a patient and a gene, it means that this particular gene is mutated in this patient. So this looks extremely valuable source of information. So potentially we can learn a lot and maybe try to predict the future of the patient, or potentially we can try to predict how this patient will respond to a particular treatment, and as a result choose the best treatment for a particular patient, which is the purpose of precision medicine, as I have already told. So these matrices can be subject of machine learning approach. And so it's kind of typical machine learning problem, supervised machine learning problems that we can try to solve using this matrix is to predict the survival of the patient. So we can try to predict, given some mutational profile, what will be the for how many years after the start of the treatment we expect a particular patient to survive. So this is a classical supervised prediction task. There are some state-of-the-art methods in this field. For example, the sparse survival support vector machine is one of the up-to-date method. So what this method takes as input, it takes this mutational matrix X. It takes a set of so-called follow-up variable Y, which tells you for how many months a patient is followed. And then you have an event vector, which is binary vector 0, 1. In this case, it represents the fact of survival or death. So if it's 1, it means that by month 10 after the beginning of the treatment, the corresponding patient unfortunately died. So the purpose of this, the analysis of this information is to predict the survival of the patients, which can be recapitulated in the so-called concordance index, which predicts for each pairs of patients, which patient, for example, for patient A and B, to predict we predict which patients survive longer than which, and for each pair of patients, we see if our prediction is correct or not. And then we can recapitulate all pairs of patients. We can recapitulate in one single measure, which is kind of average level of our prediction, or average accuracy of our prediction. What is important to understand, to keep in mind that if our prediction is a 0.5, it means a random prediction. So in this case, without any application of machine learning, if you just randomly threw a coin and we decide a prediction. So it's very bad example of very bad prediction. So 0.5, it means that we don't literally predict anything from the data that we have. And what is interesting that if we apply directly as a state-of-the-art machine learning approach to this data and to this problem, we indeed for these are the different cancer types which you can analyze and for which sufficiently big tables exist like this. We can see that indeed we literally predict at the level of random prediction, which is very disappointing. So there is somewhere a problem. Either these data do not contain the necessary information or we do something wrong. Instead of doing supervised prediction, we can apply so-called unsupervised machine learning approach to this kind of data. For example, we can apply, again, state-of-the-art method, non-negative matrix factorization, to try to cluster the patients. So having in mind that probably we can split these patients into homogeneous groups, and maybe for each particular group, we can do this prognosis and diagnosis better. So when you apply clustering, this is so-called consensus matrix of clustering, which shows how many, it's a square matrix, and it shows what is a fraction of pairs of patients which appears in the same cluster when you repeat your clustering many times. And when you apply clustering to the data, in principle, you hope to see this kind of picture after the clustering, such that you have more or less balanced means of approximately comparable size of clusters in the data. While when you apply these state-of-the-art methods to the mutational matrix, you obtain something which is completely unsatisfactory. So you have a situation of hugely unbalanced clusters when you have an existence of one super cluster of patients which combines most of the patients in one cluster, and then you have very few clusters containing one or two patients. So this is not something that you would expect from a good clustering algorithm. Again, it means that there is a problem somewhere, and unsupervised clustering is not directly applicable to this kind of data. So if you try to understand what's wrong with this data type and why we cannot analyze it in the way we usually do it with other data types, is you realize that there are two factors which can explain such a behavior of machine learning method. So the first fact that you observe that if you try to quantify the overlap between mutations in different patients, you see that it's very small. So this matrix represents how many mutations are common between each pair of patients. And you see that for most of the patients, this value is close to 0, 1. So we have only very few mutations which are common. And the other observation, which also might impact the results of application of machine learning algorithms, that you can see that the distribution of total number of mutations across the patients is very heterogeneous. So you see a small fraction of patients with a very large number of mutations, and you see also many patients with relatively few mutations. And this distribution follows more or less exponential law. So, which means that our initial data is extremely heterogeneous, and probably we should first try to compensate this heterogeneity and make, and only after this apply machine learning methods. So one of the idea in this direction was suggested by the group of Trey Edeker 7 years ago, and this idea is called a network-based stratification. So this is where the biological networks comes into the game, and this is where they can help solve this machine learning problem. So what is the idea? So here you have a representation of a biological network in the form of a graph. Of course, it's a kind of cartoon representation. Then you look at patient genotype 1. So you see which genes in this complex network are mutated in one of the patients. And the fact of having mutation in this patient is shown by a yellow node. You can see that these mutations are somehow distributed across the network. Then you have a patient 2 which have another genotype, and you map also mutations on top of the biological network, and you see how they distributed. Then in green you show those cases where there is a common mutation in both genotype 1 and genotype 2. And you can see that there is only one single gene in this network which is mutated in both genotype 1 and genotype 2. Okay, so this is a fact. This is very rare, and this is why it's difficult to compare genotype 1 and genotype 2, because the genes are mutated completely differently. But we can reason in the following way: that when a gene is mutated, probably not only the function of this gene is affected by the mutation, but also since it is involved in some biological process in which proteins interact, if this gene function is compromised, maybe the effect of the mutation is spread on some neighborhood in this network. And the idea is to kind of describe the effect of action of a mutation on some neighboring genes in the network in such a way that, let's say, this effect would be quantified not by 0, 1 values, but it will be quantified in a continuous fashion, and it will peak where the mutation happens, and then the effect will decrease gradually to some neighborhood. So this is an idea of the approach, and you can see that here the effect of the mutation is shown by different colors now. But as a result of such a trick, you see that there is the number of nodes where you have, let's say, non-zero effect of the mutation measured, it becomes much bigger, and potentially you can conclude that this region of the network, which represents some particular biological function of an organism, is affected commonly affected in genotype 1 and genotype 2. Okay, so in this method, in this paper, which was quite famous some years ago, there was an algorithm suggested, which was called network-based stratification. I'm going to explain how it works. And it was shown that after you apply this trick, for example, the clustering algorithm starts to work much in the way that we expect it to work. So it produces more balanced clusters, and also you can, so in this way, you can stratify patients into meaningful clinical groups. So how this method works? I'm not going into the very detail of the method. I just give you some basic ingredients of it. So in the beginning, we have a matrix X, which we would like to transform using this NSQN method, which stands for network smoothing plus quantile normalization. Then we have a matrix A describing the structure of the network. So it's so-called adjacency matrix. In this matrix, you have 0 and 1s at the intersection of columns and rows. And if you have 1, it means that two genes in this matrix, they are connected by an edge. Then you have a D, which is a simple derivative of this matrix. And you have an alpha smoothing parameter. Then the whole idea that you describe your mutational profile by the vector x0. So this is a binary vector which has ones at those nodes of the network which are mutated in a given patient. And then you apply this simple iterative formula in which you iterate the spread of the mutations on the neighborhood of the graph by the process which can be called random walk, which in the end produce some non-zero values on the rest of the network, such that those nodes which are located in close neighborhood of the nodes A and D, which are mutated protein, which represent mutated genes in this case, so they obtain high values, and those nodes which are located far from any mutated gene, they obtain smaller values. So the intensity of the color here is proportional to the quantified effect of mutation on the members of the network. So this is a whole trick, which is summarized here. So in the beginning, you have your binary valued matrix representing mutations. So you take each column one by one as a binary vector, you project it onto the network, then you apply network smoothing. As a result, you obtain a vector with many non-zero values, and then you stack them again into the matrix, which in this case contains values between 0 and 1. It's a continuous valued matrix. Okay. Then an important step in this approach is to apply quantile normalization. So in statistics, quantile normalization is the way to make a distribution of values in each observation identical. So here an example, for example, it's each box plot here represents a distribution of values in a particular observation. For example, here we have something like 10 observations, and you see that the distributions and the mean values and the variance of each distribution are slightly different from one observation to another observation, which might represent certain biases in your measurement. So in some situations, it has advantages to make these distributions identical, and quantile normalization is one way to achieve this. So here, for example, the same data set applied, transformed after the application of quantile normalization. I'm not going to explain how quantile normalization works. If you want, you can watch this YouTube video and to learn what is this approach. But this is what we tried on mutational data to predict the survival of patients and to compute the concordance index. We applied this approach on 8 cancer types. So these are names for different cancer types. For example, BRCA means breast cancer, and LUSC means small cell lung cancer, And when we applied such transformed mutational matrix, we see that we improve a little bit. So for example, let's take example of this lung cancer. So as once again, 0.5 is a random prediction of the concordance index. So we see that this is what we saw before. So the prediction was not that good for lung cancer when we took the original mutational matrix. Then here we predict just take into account the number of mutations. So we simplify the task. We just compute one single number representing the mutational profile. And we can see that we also predict not that great. Then when we apply only network smoothing without quantile normalization, we don't predict that much differently from the initial data. But when we combine both idea of network smoothing and quantile normalization, we see that we have a improvement in the performance. Not huge improvement, but noticeable, statistically significant improvement. And then, so it's the same situation we have for another cancer type, and again, quite disappointing. Well, that's a disappointing observation, but for other cancer types from these 8 examples, we didn't see improvement of in terms of application of this network smoothing. But well, this was our result. What we learned from this approach is that application of quantile normalization is an essential step. And also what we did, we tried a simplified method of network smoothing in which instead of repeating these iterations many times, we did only one iteration. We called it a simplified NSQN method. And I mean, to our surprise, only considering first neighbors in the network was already enough to achieve the same performance as a complete method. Which means that we don't have to consider larger neighborhoods in the network of genes larger than distance 1. So being inspired by these experiments, we decided to introduce even simpler approach, which we called NetNorm, standing for network-based normalization of mutational data. And in this approach, we convert a binary mutational matrix into another binary mutational matrix, but such that the number of mutations in each molecular profile would be the same. So it will be equilibrated in terms of the number of mutations, unlike the original matrix. So what we do, we define, so to achieve this, we define a so-called target number of mutations, K. So in this cartoon example, we use K equal to 4. And then we split all patients in our collection into those patients which have less than K mutations and those patients which have the more than K mutations. And we treat them differently. So for those patients which have less than K mutations, for example, we have a mutational profile with only 3 mutations, here, here, and here, in these 3 genes. And what we do, that for the rest of the genes, we count the number of mutated neighbors. And you can see that, so yellow and red and this reddish point are only those genes which have a mutated neighbor. So yellow, for example, yellow genes have 1 mutated neighbor. And this red gene, it has 3 mutated neighbors. Then we rank the genes accordingly to the number of mutated neighbors, and then we add a so-called proxy mutation, which means it's a fictitious mutation or pseudo mutation on those genes which have the largest number of mutated neighbors. And as such, we complete the number of mutations in this profile up to K. If we have for a patient, we have number of mutations is more than K, we do the following trick. For each mutated gene, we compute its degree. So how many neighbors it contains at all. So by such, we distinguish those genes, those mutated genes, which are located somehow, which are central to the network, from those genes which are peripheral to the network. For example, these mutated genes are quite peripheral to the networks. And we start to remove mutations starting from peripheral, from the less connected genes in the network. And as such, we achieve to the situation when we have only K mutations remaining on the network. Okay, so what is the reasoning behind this approach? The reason is the following. So here we add mutations to the so-called hubs of the network. So those with genes in the network which are connected to many other genes. And the rationale is the following. If among the neighbors of a gene, highly connected genes, there are many mutated genes that we observe with mutations, that probably the function of this gene is also compromised. And this is why we mark it as with a pseudo or artificial mutation. The reasoning here is that if we observe mutation on a gene which is very loosely connected to the rest of the network, probably the mutation in this gene doesn't affect the functioning of many other genes, and probably it's a so-called passenger mutation, which is mutation which happened by random chance, and it doesn't contribute to the dysfunctioning of a biological cell. So in other words, this network-based normalization, it normalizes the mutation matrix using so-called guilty by association principle, which consists in the fact that, for example, if in Facebook network, if your Facebook page is connected to your friend pages, and each of your friends is characterized by a particular feature, because, for example, he or she likes watches of some science fiction movies, then you can suspect that you, being connected to many such friends, also like to watch such science fiction movies. So this is what we call the guilty by association principle. So great. So we had the idea of this method, then we applied it again for predicting survival of the patients from mutational data, and we were quite satisfied. So in those cases where network smoothing had some effect, this NSQN method had an effect, we saw that in these cases, network-based normalization that we introduced, which is a simpler method, allowed to even more improve the performance of the prediction. In those situations where we didn't have good any improvement from the application of network-based smoothing, also we didn't have a noticeable improvement in the prediction, which means that probably in these cancer types, we should use maybe different types of information to predict the survival. Also, what was interesting is that we tried to combine these mutational data to predict the survival together called clinical data. I already mentioned to you that we have a certain clinical information such as tumor stage and tumor grade. And we can also use this information to improve our prediction. So what we showed that by applying this network-based normalization of mutational data, we can achieve better performance than using clinical data alone, or combining the raw mutational information with clinical data. So in other words, the type of normalization of mutational data that we suggested, which uses the notion of biological networks, allows to use a combination of clinical data and mutational data together, and it improves the prediction in the end. So by this, I'm going to stop with a simple conclusion that using biological networks can be helpful in improving analysis of molecular data, which otherwise are difficult to use in the standard machine learning approaches.